correct now.
So why did somebody now come to microbiology?
If that person was not correct.
Hello, good morning. Are we here?
Nobody is there.
Nobody's there.
Yes.
Huh?
Yes.
So check that link. I tell you, see. What?
Is it talking
This boy, get issues. Hello, good morning. Am I being heard? Hi. Welcome to Biotechnology and Genetic Engineering class. Good morning, everyone. If you can hear me, just say yes so that I know you are right here. This is going to be a very brief one, and we're just going to move very fast because I just ended another revision class and actually getting tired. But let's do this. So if you are here and active, say yes so that we'll move as swiftly and as fast as possible. Do I have a response there? It's full here, network is. Okay, I began this aspect of the course talking about biotechnology, and we said that the biotechnology can be described as a range of scientific techniques, including genetic engineering, that are used to create, improve, or modify plants. So for biotechnology, the key words, like I mentioned, is that they are used to create. Let me do that in red. They are used to create, all right, improve, and what? Modify plants, animals, and what? Microorganisms. And all of this is actually for the benefit of humans. So if you know these keywords, you pretty have covered what biotechnology is. They are keywords, a range of techniques, and these techniques include genetic engineering. As we move right along, you'll see that genetic engineering is in phases, and it has different aspects: genetic splicing, genetic modification, and all of that embedded in it. And all of that is actually used for for the benefit of man, and they use it to create, to uh, improve, and to modify plants, animals, and microorganisms. Alright, so that's pretty much what genetic engineering is. Okay, so we went about to talk about the brief history of biotechnology. As as far back as the 20th century, biotechnology actually began, and it brought industry and agriculture together. It brought industry and agriculture together, and again during the World War One. This is just a brief history. I just try to itemize it so that it does not look clumsy and jam packed. So you could just have it as bullet points like the way I gave it here. Okay, biotechnology at the beginning of the 20th century. So as far back as the 20th century, biotechnology began. That's key to note. All right, and it brings industry and agriculture together, especially the medical industry. Then during the World War One, fermentation process were developed, okay, and which produced acetone from starch. So the first um, fermentation process occurred during World War One, and it produced acetone from starch and paint solvents, which solvents for rapidly growing automotive industry. Then World War Two came, and in World War Two, it brought the manufacture of penicillin. Okay, this is key for any molecular and biotechnological process. All right, production and manufacture of penicillin occurred during the World War Two, and biotechnological focused in pharmaceuticals. Thus, the Cold War years were dominated by work with what microorganisms in preparation of bio warfare. Of course, bio warfare is warfare that we see today, like what we had with the recently abolished COVID-19, are all forms of biological warfare, whereby people want to eradicate the population of the world using biological means, okay, creating vaccines and all of that. So biotechnology is one of the powerful technology of the 21st century. So it didn't end with the 20th century. It walked into the 21st century, and it has revolutionized the life sciences that has grown into separate discipline. Separate discipline is a key word here. All right. So this separate discipline encompasses cell biology, immunology, okay, molecular biology, the recombinant DNA, and bioinformation, in which you have bioinformatics. All right. So if you are just joining in, what we've said so far is that we began from biotechnology and how it is described. Okay, and in saying that, I said a range of scientific techniques, and all of those range are grouped under the word genetic engineering right here. And we said that this genetic engineering, they are used for three main things: to create, to improve, and modify plants, animals, and microorganisms. And the major focus in doing all of this is for the benefits of man. If you are able to do this, you have been able to perfectly describe what biotechnology is. Okay, then we went about talking about the brief history, and we are able to highlight in bullet point format how the brief history occurred right from the 20th century into the 21st century. And right here we are saying that biotechnology is one of the most powerful technologies of the 21st century, that has revolutionized life sciences and has grown into separate disciplines. Okay, this should be separate disciplines that encompasses a number of disciplines. So, okay, I think that was correct. So, it has revolutionized into separate discipline that encompasses a number of disciplines, and these disciplines are, all right, that it encompasses are cell biology, immunology, molecular biology, recombinant DNA, and what bioinformation. All right, so biotechnology is currently being used in many areas, which includes, that's many specialization areas, which includes agriculture, bioremediation, food processing, and what, energy production So these are the disciplines that it encompasses, and these are the focus specialization areas that it mainly covers. All right, agriculture in particular, bioremediation, food processing, energy production, all right, is still used in this area. Okay, so we just move ahead. So you can't talk about biotechnology without looking at the living organism, because basically what biotechnology is good at is creating, modifying living organisms, which includes plants, animals, and microorganisms. All right, so it's now key to note that living organism could be either unicellular, that biotechnology uses, or multicellular. When we say unicellular, it means it consists of only one cell, and when we say multicellular, it consists of many cells. Now, bacteria and bakeries are examples of unicellular organism. That's organism that has just one cell. Why man and tilapia fish, of course, other examples you could mention, monkeys, rats, name it, are examples of what, multicellular organisms. Now, the difference between one organism and another is the encoding of the genetic material. From this point is key, because we are now diving into the biotechnology proper. You can't talk about biotechnology without talking about the basic genetic material called the deoxyribonucleic acid, your DNA. Okay, so your DNA is key, is a very key factor in looking at biotechnology with respect to genetic engineering, that's genetic modification and manipulation of living things. All right, so the living things that are manipulated, like we earlier saw, could actually be living unicellular or multicellular. Now, the difference between these organisms among the living things, either plants, animal, or microorganisms, is that, or is the DNA. How does their DNA looks like? How is it encoded? And what is the nucleotide basis? And what is the complexity and what they can do? So DNA is the main information carrier molecule of the cell. Okay, so we have 4 different nucleotides that are grouped into 2 types. Basically, we have the purines and the pyrimidines. And in looking at the purines, we have the adenine and the guanine as nucleotide bases that are associated with the purines, and while we have pyrimidines, we have cytosine and thymine as nucleotide bases that are associated with the what, pyrimidines. And these purines and pyrimidines actually form the DNA molecule structure. That is also very important to note. Okay, they form the DNA molecule structure. And right here we now saw the specific base pairing of these nucleotides, which are the specific base pairing of the purines and pyrimidines, which includes the adenine and guanine and cytosine and thymine, where you have cytosine always base pairing with what? Guanine, and thymine always base pairing with what? Adenine. Now, the base pairing of nucleotides in a DNA molecule is what you see in this structure right here, that begins from the 5 prime to the 3 prime molecule. Let me change the color here for emphasis. So this is the base pairing that you see right here. Okay, so it begins from the 5 prime to the 3 prime, and the 5 prime end is what starts it. So this figure here shows this base pairing. Oh my god. So it starts this base pairing. So we say that the end of the nucleotides are marked either 5 prime or 3 prime. And of course, what is 5 prime or 3 prime has their chemical reasons in the numbering, and that is actually based on the OH groups of the sugar ring. So by convention, DNA is usually written from the 5 prime left end to the 3 prime right end, with the coding strands at the top. So you have this, the 5 prime to the 3 prime right here, the 5 prime to the 3 prime, and these are the coding strands at the top, which includes what? The bases, your cytosine, guanine, thymine, adenine, guanine, and all of these have their complementary base pairs that they pair in the reverse direction. Okay, so 2 of such strands are shown above. 2 of such strands that are shown above attempt to be what? Complementary. Like I said, when you have these strands, you have their complementary base pairing. So automatically when you have cytosine guanine, you know that the complementary base pairing in the reverse direction from the 5 prime to the 3 prime end, in the reverse direction from the 5 prime here to the 3 prime here, we move in this direction, and in the forward reaction it moves from the 5 prime here to the what? 3 prime, and you have this basis. And like we know that in the base pairing, guanine we always what? Base pair with cytosine, and thymine we always base pair with what? Adenine. So you could actually like have a schematic process in which you are able to put up a base pairing of nucleotides in a DNA molecule for illustrative purpose, and you are able to properly produce their complementary strands. Alright, so we're moving very fast. We say that it's also worthy of note that such interactions are individually weak, and 2 longer complementary polynucleotide chains meet, they tend to stick together because they are composed of weak bonds, and when they meet, they stick together. Alright, so we move forward quickly. So apart from those bases that we saw that the DNA are also made up of sugar phosphate backbone. So individual of those bases, they are made up of sugar phosphate backbone, where you have the pyrimidine and the purines, and we have the sugar phosphate backbone. So this is a structural representation of what we have just seen earlier, and nucleotides is shown here to be the building blocks of what the DNA molecule. All right, so moving forward, okay. All right, so we have the three-dimensional structure of the DNA molecule, the 3D structure of the DNA molecule, as the next thing we saw. And we saw that the three-dimensional structure consists of a long helical strands that are coiled around the common axis. So um, worthy to note here is that we've shown you the DNA molecule in different formats. So I'm having the phosphate sugar backbone, and we have the base pairing of nucleotides, okay, in a DNA molecule, and of course with the three-dimensional structure, all of a DNA molecule. Why? Because the DNA molecule is key. In fact, is the fundamental element that is needed for any genetic modification, manipulation, or engineering process. So your 3D structure, it consists of the long helicals that are coiled around. Again, you see your guanine here. Again, you see your guanine right here, base pairing with cytosine, and thymine base pairing with what? Adenine. And you see your sugar phosphate backbone like we saw structural wise, and you see your base pair. So this contains the other two that we saw in a double helix form. So DNA is actually packaged in chromosome pairs, which are strands of genetic material, one coming from what? Each parent. All right, so we just move on quickly. All right, so this is a schematic diagram of what the chromosome look like, and the genes. So the genes which control an organism's characteristics are located on specific segments of each chromosomes. All of the genes carried by a single gamete is known as what? The genome. This is also very key. Alright, so we saw that this DNA are in chromosome pairs, and the structure of the chromosome pairs is what is shown right here, having the chromosome and a segment of it coiled around each other, that contains what? The genes. And worthy to note is that the genes is what controls an organism characteristics. The genes right here is what controls an organism's characteristics, and they are located within the specific segment of each chromosomes. So all of the genes carried by a single gamete is well known as what? The genome. A single gamete is a representative of each of all of the chromosome pairs. So we went ahead to see the different genome sizes of quite a number of different organisms as shown in the table below. So we looked at the bacteria having, of course, a unicellular organism or chromosome. We looked at yeast, we looked at the worm, and all of these we looked at their approximate genome sizes in base pairs. So that's pretty much of this slide. Now we we came to know that the genome sequence is now available as a direct method to help identify specific genes. we said so earlier that genes are located in specific areas of the chromosome known as what the genome, these specific areas, and you can identify it either by direct method to help identify genes, or what, by indirect method. And the indirect method is actually through the use of genetic markers. Very key, through the use of genetic markers. Most times, research process we use this indirect method. And so, what is genetic marker? We noted that a genetic marker is defined, or can be defined, as a gene whose phenotypic expression is usually easily discerned and is used to identify um, an individual or a cell that carries it. Charge. As a cell that carries it, or as a probe to mark a nucleus or chromosome. Right here, you could simply see two ways to attack or know what a genetic marker is. So, genetic marker could also be defined as what, a chromosomal landmark or allele. So, it's expected that at least you know one of these genetic marker definitions and what they do generally. So, you could know it as a gene whose phenotypic expression is usually easily discerned and is used to identify what, an individual. So the basic keywords that you will see in each of them is that they are used for identification to identify an individual or a cell that carries it, or as a probe that mark a nucleus, chromosome, or locus. Now, genetic marker could also be defined as what, a chromosomal landmark. It's like a sign. A landmark gives a direction, it gives a identification, it gives an indication, okay, or allele that allows for the tracing of what, a specific region of the DNA, or as a specific piece of DNA with known position on the genome. So that's what genetic marker is about, and we looked at the different terminologies that could be used. Okay, so we saw that genetic engineering, all right, recombinant DNA technology, all right, or genetic modification or manipulation, and gene splicing. They are all terms, they are all terminologies, all right, that could be used in what that could be used or applied to direct manipulation of what, an organism's genes. So I could use any of them conveniently. They all meant the same process. Genetic engineering is one, recombinant DNA is two, genetic modification and manipulation is three, and gene splicing is four. They are all terms that can apply to the direct manipulation of an organism's gene. So, genetic engineering actually uses the techniques of molecular cloning and transformation. Okay, it uses the techniques of molecular cloning and transformation to alter the structure and characteristics of gene directly. Okay, so that is also very key. How it is used, it uses techniques of molecular cloning and transformation. Why? You are actually changing, creating, or applying genetic technology, so techniques. So genetic engineering have been applied to various industries. Principal of them are the industry of medicine and agriculture. Okay, and they have some recorded successes. Okay, and examples of these um, applications of genetic engineering is that they have been seen in the production of synthetic human insulin using modified what bacteria. They have also been seen in the production of erythropoietin in hamster ovary cells. They have also been seen in the production of new types of experimental mice, such as the onco mice. And I said, what is onco mouse in class? You remember? And we said onco or oncology signifies cancer, the study of cancerous cells. So onco mouse can be said to be what? Cancer mouse. And this type of mouse are specific for what? Research purposes. They also can be seen to be used for the production of what? New strains of crop plants with beneficial qualities. You can recall the examples that we looked at elaborately in class, where we talked of having qualities of desired traits and qualities in different plants, and with biotechnology you can actually merge the two plants and have both traits or your desired traits in both plants all growing in one. Biotechnology can successfully do that for you. Okay, so we just move on very fast. We now got to the isolation of DNA. Now, like I said, DNA is key. So if you want to begin modification, you want to create new things, you want to uh, manipulate and do some genetic engineering, you will basically need to bring out the DNA to play with it. And to be able to do that is what we meant by what? Isolating the DNA. So what is DNA isolation? That's DNA is usually found in the nucleus of cell. So to isolate it, you need to get to where it is located to be able to bring it out. And you need to know how to get to that location and to bring it out. So DNA is usually found in the nucleus of cells, and that's basically for the prokaryotic cells. And we say that cells are the fundamentals of life. This should be the fundamental unit of life. Let me quickly do that correction. Cells are the fundamental unit of life. So all organisms are made up of cells. All organisms, as long as they are living, they are made up of cells. And these organisms could either be prokaryotes or what? Eukaryotes. Whatever the case, that is, whether prokaryotic or eukaryotics, the DNA is usually found located either freely in the cytoplasm, which is the case of prokaryotes, or in the nucleus, which is the case of what? Eukaryotes. So prokaryotes, in prokaryotes, we have the nucleoid consisting of a bacterial DNA and is enclosed within a membrane. And I gave a schematic diagram of what the nucleus looks like and how it is embedded within the plasma membrane, and how it's embedded within the membrane. And we saw that the DNA, which it can also be found in the nucleus, and we also saw a schematic diagram of what the eukaryotic um, cell looks like, and we saw where the nucleus is located. And in driving at the isolation of DNA, you need to break through all of these processes and get right to your target, which is what? Your nucleus. All right. So other consequence, so in giving you this structure, is not to dwell on prokaryotes and eukaryotes. This is not microbiology, but to let you know that they contain important material for genetic engineering called the DNA. And if you must modify things to suit any purpose or make things better, you need to isolate the DNA. And in isolating it, you need to know how the cell structure that you are looking at is. Whether it's prokaryotes, whether it's eukaryotes, you need to know how to get to the location of the DNA. and that's why all of this is shown. So other constituents of the nucleus includes what? The ribonucleic acids, which is your RNA, your proteins, your carbohydrates, and of course your lipids. Thus, when isolating your DNA, the first necessary step is what? The breaking of the cell, which is the disruption of the cell. And the purpose of disrupting the cell is really to release the nucleoid or the nuclear material. So you are releasing the nucleoid when dealing with prokaryotic cells, or the nuclear material when dealing with what? Eukaryotic cells. Right. Okay, so we now look at if we need to always bring out the DNA. So what is DNA isolation there for? And we said that DNA isolation refers to the process of extracting DNA from a cell in a relatively pure form. A relatively pure form is a key word here, because if you have to do anything genetic, you want to do it in the purest form as much as possible. You don't want to carry out a research that has contaminations either with lipids, proteins, nucleic materials, and all that. If you isolate the DNA, you want to know that this is the DNA of whipstar rat, this is the DNA of tilapia fish. You want to be sure it's in the pure form. So it therefore involves the separating it therefore involves separating DNA from other cellular components such as what? Proteins, RNA, lipids, carbohydrates, and the rest of it. Then you have, is a routine procedure. So therefore, if you have to do this as in genetic engineering process, DNA isolation becomes a routine procedure to collect DNA for subsequent molecular and forensic analysis. So DNA isolation is very key to what? Genetic engineering, and it's important you know what the DNA isolation is. So we now look at the steps involved in isolating this DNA or extracting this DNA. And we saw that the different steps. We saw that DNA can be isolated by placing the cells in a tube containing a special solution, normally called what? The cocktail. That special solution is called the cocktail because it contains other chemical materials that is needed for what? The isolation of a pure form of the what? DNA. How it can be isolated mechanically or what? Chemically, by breaking up the cells. Okay, so the cell is broken up. So therefore, we look at the specific steps in what? The DNA isolation, which is as follows. Number one is breaking the cell open. That is pretty your beginning point. You have to lyse the cell, you have to break it, you have to disrupt it. So breaking the cell open is commonly referred to as what? Cell disruption, or what? Cell lysis. And the purpose is to expose the DNA within the cell. This is commonly achieved by grinding or sonicating. You can either do that with your mortar and pestle, you could also do that with your blender, you could also do that with your sonicating machine. Whatever it is that can break the cell of interest is very key. So it's achieved using different procedures. Number two in the steps of DNA isolation is removing membrane lipids by adding a detergent such as sodium dodecyl sulfate, your SDS. You also have cetin-methyl ammonium bromide, which is your C-tab, or your mixed acyl trimethyl ammonium bromide, which is your M-tab. These are solutions that act as detergent in removing lipids from the isolated DNA. So you are in the process of actually purifying the DNA that you have extracted from the cell by breaking it up. So this causes the cell to release its content into the cocktail containing enzymes, chemical, and such. Remember that in step 1, um, the first step is that is um, it can be isolated by placing the cells in a tube containing what a special solution called cocktail. So when you pull out these cells and you use your SDS, your CTAB, and your MTAB your MTAB to remove lipids, this will actually cause the release of its content into the cell, release of its content into the cocktail rather, containing enzymes, chemicals, and salts. The third point or the third step in your DNA isolation is that you remove the proteins by adding a protease. A protease is an enzyme that we remove protein, okay, that catalyzes the removal of what proteins. So it's always an optional process, but it's always almost done in during research. Why? Because researchers want to obtain a purest form of DNA as much as what possible. Then you have the fourth step as precipitating the DNA with an alcohol, usually your ice cold ethanol or what your isopropanol. Alright, so when you precipitate this DNA, having removed some proteins and removed the lipids, you are getting closer to the purest form as much as possible. Then you do a wash with your ice cold what ethanol or isopropanol. Since the DNA is insoluble in these alcohols, it will aggregate or precipitate together, giving a pellet usually at the bottom of the tube, okay, upon centrifugation. So you can centrifuge at a specific revolution, and when you do that centrifugation, you see that your DNA, since it's insoluble, it's not soluble in alcohol, it will gather together, aggregate, or precipitate at the bottom of the tube as pellets. So you can just neatly remove your supernatant, and there you have your DNA. So these steps also remove all alcohol soluble salts. Alright, so okay, this is pretty it, and we say that the DNA extracted will also exist as long strands like we saw earlier that forms a mucus like globe within the solution. So as the DNA is released along with other compounds like your lipids, proteins, carbohydrates, the DNA therefore needs to be separated from others. So there are other steps right down the way, but as much as possible, because of the purpose of this class, which is to revise for your exam, we want to keep this within the space of what we had lectures for in class. And with this, we come to the end of this segment. I am now open for questions. Thank you. So you please type in your questions if you've not done that, so that if you are pretty good, we could just move right along. Okay. Do you have any questions? I want to know, did they hear me at all? Okay, so any question yet? Is there any area that is not clear to you, so that I can allow you have your night back and you rest a bit and wake up, and like these live lectures will still be up, so you could easily just like a form of revision look at it. So in the absence of any question, I do you have questions? Question, please give me the question. Well understood. Okay, so do you have question? Yes, ma. Okay, somebody said it's okay. Alright, thank you. Well understood. Thank you. Alright, drop your questions. Well understood, no questions. Okay. Thank you. I am receiving your chat comments. No questions. That means it's pretty straightforward. And with that, I want to congratulate you ahead. Have a good rest, and come back maybe in the morning to look at it. At least you are assured of tackling um, about 3 questions of your complete course from this lecture. So I want to say congratulations in advance. And with this, we close the live lectures for today. Wishing you great success in your exams. Bye.